first of synthetic embryos in the world and a revolutionary feat that has circumvented the need for sperm, eggs and fertilization. Scientists from the Weizmann Institute in Israel have found that mouse stem cells could be designed to self-assemble in embryo-type structures with an intestinal tract, the beginnings of a brain and a beating heart. Known under the name of synthetic embryos because they are created without fertilized eggs, living structures are expected, in the short term, to stimulate a more in-depth understanding of the way in which organs and tissues are formed during the development of natural embryos. But researchers believe that work could also reduce animal experimentation and finally pave the way for new sources of cells and tissues for human transplantation. For example, the skin cells of a leukemia patient could potentially be transformed into bone marrow stem cells to treat their condition. Remarkably, we show that the embryonic stem cells generate whole synthetic embryos, which means that this understands the placenta and the yellow bag surrounding the embryo, said Professor Jacob Hanna, who led the effort. We are really excited by this work and its implications. The work is cellular. Last year, the same team described how they had built a mechanical uterus that allowed natural mouse embryos to develop outside the uterus for several days. In the latest work, the same device was used to feed mouse stem cells for more than a week, almost half the gestation time for a mouse. Some of the cells have been pre-treated with chemicals which have activated genetic programs to develop in placenta or yellow bag, while others have developed without intervention in organs and other tissues. While most stem cells have failed to train embryo-type structures, about 0.5% have combined in small balls that cultivated distinct tissues and organs. Compared to natural mouse embryos, the synthetic embryos were 95% the same in terms of internal structure and genetic cell profiles. Regarding scientists, the organs that were formed were functional. Hanna said synthetic embryos were not real embryos and did not have the potential to transform themselves into living animals, or at least they did not do it when they had been transplanted in the uterus female mice. He founded a company called Renewal Bio which aims to develop human synthetic embryos to provide tissues and cells for medical conditions. In Israel and many other countries, such as the United States and the United Kingdom,